Shalom everyone. So I won't be long, I will be actually very short. I just wanted to welcome everyone. This is an exciting actually moment. This is the first time we do the seminar. We opened the auditorium in the new Goodman building. We're still struggling to move in with our move into the building with numerous issues all over the building, but this is actually a nice gem that is fully functional, and I'm very happy to open the weekly seminar series. And without further ado, our first guest is Randy, which will be presented by Yoram. So, thank you.
Okay, so it's a great pleasure to have as our first speaker of the year and of this new auditorium, Professor Randy Blakely. Randy Blakely did his undergraduate studies at Emory University in philosophy, physics, and chemistry. He then did a PhD at the School of Medicine in Johns Hopkins. Since 1995 to 2016, he was a professor of pharmacology and psychiatry at Vanderbilt University, and since 2016, he is a professor at the School of Medicine in Florida Atlantic University and the founding executive director of the new Brain Institute of Florida Atlantic University. In recent years, much of his work has focused on the genetic underpinnings of neuropsychiatric disorders. He has more than 300 publications, many of them in leading journals such as Science, Nature, PNAS, and others, and he holds dozens of patents. He receives many prizes and honors, too many to list completely. I'll just mention that some of them were given by the American College of Neuropsychopharmacology, the Brain and Behavioral Research Foundation, the National Institute of Mental Health, and other associations and fellowship and societies. And he is an elected fellow of the American Academy for the Advancement of Science and the National Academy of Inventors. So thanks for coming, and you can see the title here.
Great. Well, thanks a lot. This is my first time, obviously, here, and first time to Israel as well. I'm sort of a representative here of my institution, my new institution, as well as to talk about my science and meet new people. I'll talk to you about our efforts to understand a bit more about a disorder that we think has many changes associated with dopamine signaling, and I'll show you one perspective on that and one way to get into it. First, because I hope that my visit will allow my university and the Brain Institute that we're developing to develop close ties with a number of Israeli universities. I want to tell you just a little bit about Florida Atlantic University because I know, since it's very young, that you know nothing about it. So let me just make a few points and then get into the science. So Florida Atlantic University is actually a collection of campuses or mini universities running up and down the Atlantic coast of Florida. My home is in Jupiter, Florida. The main campus, his home is in Boca Raton, which interestingly enough is 80% Jewish. So it has a lot of ties back to Israel. It's a very pretty place. My lab is 5 minutes from the beach, which means sometimes 5 minutes from a hurricane. So fortunately, the hurricane came up here this time. But my first 2 years, we had 2 hurricanes came in, and my lab was very happy that we moved from Tennessee down to Florida. But it's a really beautiful place, and I hope you will have a chance to come. There's 30,000 students total across these campuses. It's most of the neuroscience is concentrated here in Jupiter and Boca Raton. And just to give you a few visual aids, so this is our home here. This is the buildings I'm in, and this is the College of Medicine in Boca Raton. It's a state university, one of the 10 state universities in Florida, and so it has a lot of professional schools as well as typical sort of graduate programs, a couple of which are in neuroscience. Now, the Brain Institute itself has I have several responsibilities as part of that. It's one of 4 major investment areas that the university has decided for the next 10 years to focus much of its resources on. So that neuroscience pillar, each pillar has an institute and an institute director, and so that was myself. And as was noted, I came from Vanderbilt University after 21 years there. We have 63 faculty-level neuroscience investigators, again, spread a bit across campuses, but mostly in Boca Raton and Jupiter. Multi-college, multidisciplinary, runs from genetics and biochemistry to cognitive neuroscience. We're responsible for establishing core technologies. This is a relatively young neuroscience community, and so we've just begun the building of core facilities for cellular neuroscience imaging, neurobehavior, and transgenic mice and the like. And so that's ongoing, but it's going very well. And then lastly, we've been building educational programs, neuroscience graduate programs, a postdoc association, and community outreach programs. So my home, as I said before, is here on the Jupiter campus, and out my window I can see Max Planck, Florida. It's the only Max Planck outside the U.S. is here, and it's really a wonderful set of colleagues there. And looking out another window, we have Scripps, Florida. So this is a partner to its home in San Diego, and is very focused on drug development and moving the discoveries from the laboratory into medication development. So I hope you'll take a little bit of time, maybe, after hearing this, to go check out our website. It's ibrain.fau.edu, and even more, I hope you have a chance to visit in the near future. And I can promise you at least beauty and hopefully some good science and conversation also. So let me get to the science part of this. I'll tell you a bit about our work looking at, as it turns out, and not where we were starting, presynaptic autoreceptors and their relevance for the control of dopamine signaling, and how brain regions that receive dopaminergic input are different in how they control their dopamine transporters. And this makes a difference when you're looking at mutations that we think can induce disorders like ADHD. So I want to first talk a little bit about attention deficit hyperactivity disorder itself, move into our genetic work looking for mutations underlying this disorder with a focus on the dopamine transporter, and then tell you a little bit about the mice that we've been working with and some of the findings from that model to date. These are still relatively new studies. The work I'll tell you about was published about 2 months ago. So some basic facts and statistics. These are numbers from the US, and I doubt that they're much different here. Attention deficit hyperactivity disorder in our country is the most commonly diagnosed neurobehavioral disorder of childhood. About 8% of kids will reach criteria for ADHD, have a formal diagnosis. There we go. Okay. I think you all are aware that we have a real problem in psychiatry in terms of defining disorders on anything other than behavioral criteria, and ADHD is no different from that. We recognize the symptoms of inattention, impulsivity, and hyperactivity, and by DSM-5 criteria, those need to be identified by age 12 or seen in subjects or by that time if they were diagnosed later. There's a male bias of 4 to 1. You see the same sort of thing in autism. And we don't understand in either disorder where that male bias is coming from. The women I know like to talk about protective factors that women have, or that males are weak in some fashion. And we really don't know, and this is an interesting facet of our research going forward, and I will show you just a little bit as we've started to enter into that discussion. So about 60% of individuals with ADHD in our country are treated with medication, and 25% receive no treatment of any form. Medication to treat this disorder is actually quite effective as a much higher much higher rate of success than, say, using antidepressant therapy does. And it may give us some clues about the underlying biology associated with the disorder. And lastly, untreated ADHD results in a higher incidence of substance abuse, conduct disorders, even incarceration. And so it's important for us to not only identify the disorder in individuals and kids, but also to do something about it, whether that's medication or something else. Now, again, back to the diagnosis of ADHD. Here are the classes of symptoms. It's essentially what forms a checklist for a physician in looking at these criteria. And usually in a group here or group here, one needs to have 6 of these, and they need to be prominent. And the actual cluster of these need to affect the life substantially of the individual, of the child, and it can't be situational. So it shouldn't be that you show these things because you've had a high sugar drink or you're in a particularly stressful environment. These are common underlying traits. So we have inattentive symptoms, which my wife says I have, like not seeming to listen, failing to finish tasks. I think I do pretty well in my world, but not at home. And losing things. So I'm sure I'll have to collect something before I go here. And then hyperactive impulsive symptoms, moving around excessively, talking excessively, blurting out answers, being, interfering with other people during the normal course of events, and even being confrontational. And these all we have at some level, and this is the spectrum, but this really has to affect the life of a child. In terms of interfering with their learning opportunities, their conduct, before ever it would reach a diagnosis like this. Was there a question?
Yeah, this is not a joke. The inattentive symptoms look like ones that we all have.
Yes, we all have.
I think most people have. So what is the standard? How do doctors create criteria here?
Well, you may be. This is a spectrum, and you may be here on this spectrum as inattentive, predominant ADHD, but you wouldn't get the diagnosis unless this disrupted your life and was compromising, particularly your educational opportunities. This is when you typically diagnose this at school-age kids. And some of these can be quite severe. So let's get into the biology a little bit of this. So, heritability in this disorder seems to be quite high, 60 to 90%. There have been genetic studies that have identified common and rare gene variation. And when you look at where they cluster, 2 prominent groups that relate to us are in neuronal development and synaptic function. There are other disorders that run in families with ADHD, and those include bipolar disorder and autism. And so individuals with autism, 60 to 70% will meet criteria for ADHD. And in case of bipolar disorder, there is a higher frequency that an individual who has ADHD will live within a family group where second or third order relative will have bipolar disorder or have earlier onset bipolar disorder. And that's suggested that there may be this underlying genetic pathways that may express themselves one way or the other depending on other genetics or environmental problems. So as I said before, most of the work actually to date has been associated with an effort to look at common genetic variation. The problem with that is that it's often of small effect, and it's very difficult to model this in a laboratory and see signals that you are willing to go after and study, and that have strong effects that you can get clear phenotypes. And so there are a few examples, however, where there are rare genetic changes, such as in Fragile X, that have large effects, and almost all of subjects with Fragile X meet criteria for ADHD as well. I'm going to tell you a little bit about rare genetic variation that's sort of moderate effect. and it leads to what we think is increased risk for the disorder. But there are individuals who have the variant I'll talk about that are would be, quote, normal. And so we just keep this in mind. It's going to be a constellation of other factors probably working with the variant I'm going to talk about, which is penetrant in the mice and penetrant in vitro. But a question about whether it rises to the level of a disorder itself is going to depend on other things.
So you were saying 60% of the 5.5 million kids are being treated.
Yes.
And you're saying that it's also comorbid with bipolar and autism.
Yeah.
Is there a change in this population in their expression of bipolar and autism? So is the treatment also effective in reducing the penetrance of bipolar and autism?
No. The treatments that are effective in the treatment of ADHD, they don't remove the core symptoms of those disorders. So there's other things on top of that. What they will do is help treat the symptoms that relate to ADHD within that population. So I want to talk a little bit about a neurotransmitter that we've been studying, dopamine, and give you a sense of why we think that there are changes associated with dopamine signaling in attention deficit hyperactivity disorder. From basic science studies going back now, 40, 50 years, we've learned that dopamine participates in circuits and modulating a number of other circuits that control our ability to detect cues in the environment and determine how salient they are, attention, executive function, reward motivation, and locomotor activity. And if one thinks about some of the symptoms that I mentioned, it's certainly easy to think about how disruption in these various units here could be contributing to the disorder. So this is the sort of the first line of discussion, which is a correlational one between what the circuits do that use dopamine and the defects that we see or alterations that we see in kids. And just on that basis, just as a segue, just to remind you of the pathways, the major pathways where dopamine is found in the brain. There's a collection of neurons in the ventral tegmental area that project into an area called the nucleus accumbens that are responsive for cue saliency and reward detection. And then a population of dopamine neurons in the substantia nigra, which project to more motor and habit areas of the basal ganglia, the striatum, and that secrete dopamine there, and that when their lesion produce Parkinson's disease. So we have two major outputs of dopaminergic neurons and some distinct symptom clusters. During the addiction process, there are transitions in the ability of dopamine signaling to be activated here versus the functions of the basal ganglia, but that will be something we can talk about later. So just to lay it out then in terms of these projections again, in a way I'll come back to, the substantia nigra, pars compacta, projecting into the dorsal aspects of the striatum are involved in locomotion and habit control and impulse control that we might think about in relation to hyperactivity and impulsivity. Then we have this ventral tegmental area projecting into the ventral part of the striatum in the nucleus accumbens. And then lastly, other areas of the mesolimbic system projecting into prefrontal cortex and involved in working memory and attention. So it may be that even though I say dopaminergic dysfunction, it may be that different individuals, perhaps along that spectrum, might, if they have a change in dopamine signaling, have it in different components of this circuitry. And still, at this point, we really don't understand that for humans, and we're trying to get at it in a mammalian model in this work. So what I'm going to talk about in terms of the molecular biology and genetics of this is a protein here shown on a presynaptic dopaminergic terminal, the dopamine transporter. So when dopamine is released at dopaminergic synapses, it acts on postsynaptic and presynaptic dopamine receptors. Its lifetime and its amplitude of its signal need to be tightly constrained, and that's carried out by this dopamine transporter, which is an active cotransport system and rapidly clears that dopamine and, and is a single mechanism by which dopamine's actions are limited. It's not degraded out here. It's actively cleared out of synaptic spaces. You can see the importance of the dopamine transporter, I think, quite well in a knockout mouse study using carbon fiber amperometry, which just allows us to sense dopamine based on oxidation of that dopamine at the carbon fiber. And when you evoke release in a striatal slice, you get a very sharp rise in dopamine levels that quickly falls back down. And as you progressively eliminate copies of the dopamine transporter gene, you can see that not only is the peak reduced, and that has to do with recycling of dopamine that doesn't happen and you don't have it to reutilize, but really impressively, the kinetics of the clearance are dramatically altered. Even here, although not blown up to make it impressive, this is an order of magnitude slower clearance of dopamine. So just losing one copy here could have fairly strong effects, and it does in mice that have the dopamine transporter eliminated. One other component of this dopaminergic system I want to illustrate for you, because I'm going to talk about its contribution to our work, and is the presynaptic dopamine receptor. So this is a G-protein coupled receptor, much like the receptors that dopamine would act on postsynaptically, but it's located here in the presynaptic terminal. It's also located back on the dopaminergic cell body. And in both compartments, it acts to negatively regulate dopamine signaling as a feedback mechanism. So in this case, when dopamine is released, it acts on these dopamine receptors. And these dopamine receptors can do three things, at least that we know of. One is to shut down the synthesis of dopamine through the rate-limiting enzyme tyrosine hydroxylase. Another thing it does is to suppress vesicular secretion of dopamine storage granules. So again, dampening release. And the third thing it does, as a transporter biologist that I'm most interested in, is it controls the level of surface expression of the dopamine transporter. And so if there's a lot of dopamine out there to clear, dopamine receptor gets a signal to mobilize those transporters, and they move to the cell surface, and your absorptive capacity increases. So three ways to get to a feedback mechanism that allows the dopamine signaling to remain in check, even though you're changing the firing rate of those neurons. All right, so here's a crystal structure of a dopamine transporter, initially work from Eric Guo's lab, and these are models produced by Cristina Fanola-Ferrer at the NIH, and it shows a 12 transmembrane domain protein with sort of a twisted barrel-like conformation structure in the plane of the bilayer. And in the middle, we have ions like sodium and chloride. In this case, just showing sodium with dopamine bound in this interior area. This is a closed conformation. So at one point, the transporter was open, dopamine and sodium bound. It closed, and then it pushed this dopamine through into the cytosol, like that. All right. So back then to the dopamine transporter in ADHD. So there have been several lines of research that have pointed to this protein and changes in dopaminergic biology in humans that we've paid attention to. One has come from PET imaging studies where we're actually labeling the dopamine transporter in individuals with ADHD. This is work from Nora Volkov's lab, where you can look at the density of the dopamine transporter in the basal ganglia of a human. And if you give methylphenidate, for example, one of the treatments for ADHD, and dopamine levels actually rise, you'll displace this ligand and you will not see the labeling, indicating that, in fact, it's binding to the dopamine receptors and is lodged in this area because of that. In this study, the subjects showed reduced levels of binding of the radioligand. It was a cocaine analog. Dopamine transporter is famous for binding cocaine and being the initial target that sets off reward responses. And so you can see this lower density, and that can either be because there's fewer transporters there, or there's fewer transporters that are available to bind the ligand. And in both cases, you get a lower signal. And the latter case can come about because there's too much dopamine ambiently there that will prevent this radioligand from accessing transporter. And I'm going to show you an example of where that latter explanation may actually explain this data better. The other major thing I think that has pointed us in the direction of dopaminergic alterations in ADHD and the dopamine transporter as a potential locus for disease-causing mutations has been the fact that the major medications that we use to treat ADHD hit the dopamine transporter. And those are methylphenidate or Ritalin, or various forms of amphetamine that now we use as Adderall or Vyvanse or those kinds of medications to help people with ADHD. And here's just showing that the Ritalin, for example, is a blocker of the dopamine transporter and elevates extracellular dopamine and brings about therapeutic treatment in that disorder. Now, in the genetic evaluation of the dopamine transporter, before I ever got into this, work had been done looking at sequence variants associated with the human dopamine transporter gene. And the major finding had been that genetic alterations in 3' non-coding region, this so-called 3' VNTR, had been found to be associated with ADHD. First, the work coming from Ed Cook's lab, and then in several other studies following this up, along with a DRD4 dopamine receptor. So actually, in ongoing work, people are trying to understand how that sorts out by subtype and by bad or good drug responses. But this 3' VNTR has stood up on meta-analysis to be related or associated with the disorder. Possibly by changing the expression of the dopamine transporter, translation of the mRNA or abundance of that level. So we took a different approach because at the time we started these studies, there were no disease-associated coding variants had been identified associated with the dopamine transporter, particularly in relation to ADHD. And we figured that if this really was an important locus for disease risk, there should be individuals out there with mutations in the dopamine transporter. And so we've been looking at this gene in a number of studies, and out of that has come these 4 coding variants that we've identified, and 3 of the 4 of these have very strong phenotypes in vitro, and I'm going to tell you about one of them today, which is this coding variant A559V, that's on the 12th transmembrane domain, actually quite far away from the binding site, but we know that these things can communicate through the plane of the membrane, and also this carboxy terminus here is a site for dopamine transporter regulation. But we found this early on in a small pedigree here shown where we found individuals who were heterozygous for this valine variant. This is a very subtle change. So, you know, you look at it, if you had a computational model and you look for disease-causing mutations, this one would never be called. It was on the background of a homozygosity of the disease-associated 3' non-coding variant. So we thought about this may be, you know, sort of a double hit, and this may be a context in which this could, in fact, rise to causality. But what's most interesting to us was at this time, many hundreds of individuals had been sequenced who had ADHD, and this had not been found. But this had been found once before in bipolar disorder by Grunhage et al. And to have 2 comorbid disorders with a rare variant showing up in the same amino acid, we thought we should pursue it a little bit. Now, most recently, another couple of individuals with this variant have been found to have autism. So I think it's pretty interesting from a limited amount of individuals who've been studied who have this. And I think the more compelling data about this particular variant has to do with the actual function of the molecule once you get it in vitro or get it in an animal. So when we first expressed this in vitro, we saw that the A559V variant had normal total dopamine levels, dopamine transporter levels, surface expression wasn't affected, transport capacity was fine, had a normal affinity for dopamine, cocaine, amphetamine, methylphenidate. And so, you know, that leads to the normal despondent reaction of the graduate student who had spent a couple of years finding this variant and characterizing in vitro, thinking that they had to start over and look for something else to do. Unfortunately, we happen to have a close colleague, Aurelio Galli, who studies dopamine transporters in a different way. Using the carbon fiber approach that I mentioned before that can sniff for dopamine, Aurelio's lab was specialist in looking at the efflux of dopamine out through the transporter. Amphetamine is an interesting substrate analog that through changes in the cell, particularly through calcium elevations, will cause an increase in the inward-facing conformations that will ultimately drive dopamine out of the cell, plus a few other changes I'm glossing over. So Aurelio used this technique to study amphetamine-induced dopamine efflux. And after having exhausted all of the measures we could, looking at dopamine going in the cell, we figured we should look in the opposite direction. And then, within a few experiments, it was very clear that what this variant was imposing was an outward leak of dopamine. You can see it here in this amperometry recording from preloaded hex cells. And you can see that the baseline is elevated compared to transfected protein that's of carrying the wild type allele. And you can tell that this is dopamine transporter or DAT mediated by simply blocking the transporters from the outside with cocaine or methylphenidate. So the transporter appears to be capable of this outward efflux of dopamine. If you take these cells and you depolarize them to sort of mimic what might be happening in a neuron, what you see is that as you depolarize the membrane, this efflux gets bigger and bigger and bigger. And there is evidence from prior studies that there's a voltage dependence on this conformational stability of the transporter. So imagine that you're a nerve terminal and you're full of dopamine and you're firing. We would predict that at each one of those epochs, there would be a leak happening of dopamine. And lastly, here is a surprise to us. We saw this phenotype. We said, okay, the nerve terminals are leaky for dopamine. Let's go back and look at the clinical record. And I bet you these people are being treated with Ritalin, methylphenidate, which is a DAT blocker. And we looked at the record, and it turns out that they're treated with amphetamine. And that didn't make any sense to us because amphetamine causes efflux. So why would you give a subject who had that drug? And what turns out is that we were falling into a trap that many people do, which is to interpret the way a drug acts based on a wild-type target. And if you have a mutant target, the drug may act in a very different way. And what you see here is that amphetamine causes this efflux of dopamine in the black side here with a wild-type dopamine transporter, but in the mutant, it actually inhibits dopamine efflux. So it acts like methylphenidate. And we think that it's lost its ability, may have lost its ability to be transported and is stuck there like a blocker. I don't think so, but that remains a possibility. But I think actually what this variant is doing is shifting conformation of critical phosphorylation site on the inside that's needed to regulate its inward-outward orientation. So our model then sort of wrapped that little segment up is that normally you have excitation-induced vesicular release of candy corn, dopamine, and that normally you have this efficient dopamine transporter to clear it out and make things synchronized so that the postsynaptic cell knows what to expect when there's a certain amount of excitation. But in the case of this mutant, what we have is this leak pathway, this short circuit, that's going to present dopamine to the target independent of whether or not this is firing. And you can imagine that, if you thought about this as like if you're the postsynaptic side and you're looking at dopamine coming in, you've got now this noisy signal. It should be precise, but now it's surrounded by this halo, this activity that you can't predict why it's there. So that's just an idea, so we have to go make some mice to see if we can understand this a bit better in an intact system. And so we did, using at that time stem cell mediated homologous recombination, and we placed this valine variant into the native locus at the appropriate site. And I can tell you that those animals just look fine. They grow normally, they reproduce fine, they don't have any overt physical changes associated with them. Now, if they had lost their dopamine transporters, the transport wasn't working, let's say that mutation was not benign at all but killed transport activity, this is what it would look like. This is what a dopamine transporter deficient animal looks like when you drop it in a cage. And I don't have to tell you which of these mice is the knockout mouse because it has profound hyperactivity. We could watch this for another 20 minutes and this picture will not change too much. But there's a problem. Although the dopamine transporter knockout mice, because of this hyperactivity, have been considered in hundreds of papers now to be a model for attention deficit hyperactivity disorder, individuals who have a full loss of this have actually now been found, loss of the dopamine transporter. And what they have is an infantile Parkinsonism dystonia. They actually get into contorted postures and cannot move. And these kids are very debilitated and often don't thrive and don't live beyond a few years of age. So that's not what these mice show. So it's important, I think, for us to continue to find more naturalistic changes in the transporter if we want to look at what happens if this does not work. And so that's why we were also drawn to identifying these mutations in individuals that had a reasonable disorder that was connected to dopamine signaling. So when we put this into the mice and we took some brain regions and we looked at it biochemically, just as we had seen in cells in culture, there's really no change in dopamine transporter levels or dopamine transport kinetics. And when the animals are in a cage and we look at their locomotor activity, you really don't see much at all. There's a little bit of hypoactivity, if anything, but this is pretty normal given the number of animals we looked at, regardless of their genotype. What they do have in terms of motor activity is a hyperreactivity, which shows as a motor hyperactivity, but it's context dependent. The biggest way, if you just reach for these animals, something that is done time and time again as their cages are changed and as you handle them, they show this what's called a darting response. And you can see the plot of the data here, that as we increase the number of mutant alleles, this shifts to the right and they become little darters in the cage. They're also very squirmy if you handle them, and it's possible just by handling to genotype these animals. Now, I'm going to skip over, just for the sake of time, some work we've done with drug responses. They show a hyporesponsivity to psychostimulants, which we now believe is due to an ambient elevation of extracellular dopamine that essentially gets in the way of that. Individuals with ADHD are actually treated with very low doses of psychostimulants compared to, let's say, what you might use if you were abusing these agents. And we think we understand a little bit now about why that might be the case, and we can talk a little bit about it. But I wanted to get into some of the other behavioral alterations that they show. And one is we were very interested in impulsivity as a trait that they might show. And what we use for that are these little mouse iPad kind of things, where with their nose they go up and touch an object on the screen, and they're trained to touch either one square or another, depending on which one is rewarded, or they can touch an object that's different from another object, and they're quite easily trainable on these instruments in a way that's not really possible for them to do if they were lever pressing. And so this is just an example, if you haven't seen this, of what happens. Can't turn it down, sorry. So the animal is getting a light as a cue, and then it hits the right. It's been trained to know to go to a particular one of these, and then so it's waiting. It's got its cue, and now it goes and touches, and now it gets its reward. And you can just set this up. you can set this up in an automated fashion, and then you can change the parameters and see how rigid their behavior is. In particular, the one that we care about as impulsivity would be in waiting. Can they wait for their reward? And so in training these animals, one thing we found was that actually the learning curves, as we train them up and then as we execute this 5-choice paradigm, actually show us that these animals have a mild but significant improvement in their ability to learn this task. And we now believe this is because they're very highly motivated by the reward given when they're training. And this is something that we found in other tests. They're willing to work harder for a reward and are more responsive to it. Now, when we explored the time delay between the cue and pressing one of these panels, now we began to get a phenotype. So what we did was we imposed a 5-second delay from the cue to the press, and if they hit it sooner than that, then they were not given the reward. There's no negative stimuli here. And what you can see in animals that are trained on this fixed delay, that they in fact show impulsivity. They have this waiting problem, and more of them fail the test than the wild-type animals do. However, if you train them on a variable task, and that by a variable delay, so what you do is you train them one day on 5 seconds. You train them the next day on 10 seconds, the next day on 2 seconds, and you just vary that around. And now you put them back on the same schedule that you started with, and now you ask them to wait. The predictability of that, of the time, is much lower, of course. And so what you find is actually these animals now are less impulsive than the wild-type animals. And I won't push this argument too far because it is a stretch, but we know from the educational literature that individuals with ADHD have a big problem with a repetitive learning experience, and they need a lot of variation to perform, but they will perform just fine. And so it's interesting that these mice show this. Now you could say, well, maybe they have just a problem with time estimation. And the reason that they screwed up in the first case is that they're just not marking time properly. And there's actually a test to do that where we look after testing them on fixed times how well that their behavior follows the expectation that they're going to hit the symbol at the right time. And you can see that when you do that and you look at the average response rate, whether it's 5 seconds or in the delay that's 15 seconds, they actually mark time just fine. But what you see again, as I mentioned, is that these animals, even though they're marking time appropriately, these guys are responding at a much higher rate now in this test. And so that's, again, an aspect of this increased motivation for a reward, that the other animals are starting to fall out and not respond very much when there's a delay, but these guys will stay in there and keep responding. Okay, lastly, I want to point out that in the model that I mentioned, there's efflux going on these transporters, but I haven't shown you any evidence of that in vivo. And I just want to show you now a little biochemical data. We're using microdialysis into the dorsal striatum. What we see is that the basal level of dopamine here is quite low, and then in a gene dosage dependent manner, we see elevation in extracellular dopamines, quite striking elevations. So their tonic level of extracellular dopamine is higher. We show impulsivity. We show some kind of reactive hyperactivity in them. And in other tests that I'm not going to show you, they seem to also have a deficit in working memory. Now, in an effort to characterize and dig a little bit deeper into what's causing this dysfunction, given this fairly subtle mutation, we went to brain slice studies to see whether or not we wanted to take the circuit apart in this case. We wanted to ask whether or not this was a consequence of development and it altered their interpretation of the rules of the game in a behavioral test, and therefore there were just changes in the dopamine signaling system that had other effects, or could we actually see this locally within dopaminergic synaptic areas. So what we did was we cut brain slices from the mice, and we looked to see whether or not they had any alterations in response to amphetamine or to 4-aminopyridine, two different ways of getting dopamine release. In the case of amphetamine, it's transport reversal, it's efflux. In the case of 4-aminopyridine, it's depolarization. This will trigger depolarization of the membrane and vesicular release. So what you see over here is that the amphetamine-induced dopamine release here is blunted. And you can see from the, if you remember, I told you that in cells in culture, the efflux of dopamine is also blunted because these are essentially acting like antagonists. So amphetamine blocks, causes less dopamine release here. This effect is also less sensitive to quinpirole, a presynaptic D2 agonist. And so you can see that the quinpirole acts to increase this signal, but not in the compared to the wild type with no drug. And you can see down in the mutant, they're non-responsive to quinpirole. So something has happened in these nerve terminal preparations to make their presynaptic D2 autoreceptor incapable of stimulating further efflux here. Reducing dopamine release like it's supposed to. It's a D2 agonist. And that was expected. We weren't surprised by that. But this was really surprising. So the depolarization evoked dopamine release was also affected. So here, just giving 4-AP to the slice, we saw, even though the content's the same, and as far as we can tell, the number of dopaminergic terminals the same, they don't release the same amount of dopamine. And we could determine that that was also D2 mediated by looking at the sensitivity of that process to quinpirole. And remember again that that quinpirole is a D2 agonist, and it should reduce the release induced by 4-AP, but it doesn't have any effect here. But if we block that receptor with raclopride, what we find is that we get now the normal dopamine release we expected. So what this is saying is that those D2 dopamine receptors that that normally can suppress dopamine release in response to 4-AP are just not working in the case of the mutant. And so why is a dopamine receptor, dopamine transporter mutation affecting this presynaptic D2 receptor? Well, our explanation for it was that in the case of the leaky dopaminergic terminals, this increased release of dopamine was resulting from this mutation and building up this sort of cloud of extracellular dopamine that would really have a profound action on this D2 dopamine receptor all the time. So instead of it being coupled to activity here, this would be a tonic elevation of extracellular dopamine giving you D2-mediated inhibition, and that's why you see this blunted 4-AP-induced vesicular release, because it's inhibiting that process even before you start stimulus. Now remember I told you that one of the effects of the presynaptic receptor is to control the trafficking of the transporter. And so it's actually a positive feedback system in this case, because normally when you get dopamine out here, this D2 receptor puts more transporters in the membrane. But that's not a good thing. These are leaky dopamine transporters. And now what I've done is I've shoved these into the membrane. And so in this particular case, this short circuit is now even worse. So that was pretty interesting, and it helps us explain a little bit about some of amphetamine's actions, actually. But I'll have more to say about this as we dive even deeper now into the different brain regions that I mentioned signal through dopamine, the VTA into the ventral striatum or nucleus accumbens, and the nigra into the dorsal striatum. So when we first looked at the dorsal striatum and actually measured surface levels of the dopamine transporter here in these Western blots in a biotinylation assay, what we found was that in the mutant here in orange and the wild type in purple, you can see there's this elevated tonic level of dopamine transporters at the cell surface. Total dopamine transporter levels are normal. If you give a D2 agonist, quinpirole, you stimulate surface expression of the dopamine transporter. But these are already elevated, and by our hypothesis, already D2s have already been flooded and are active. And so you can see that you now do not get any further activation and perhaps even get a little bit going in the opposite direction. We can talk about that, but this wasn't significant in this particular experiment. We think that this is in fact D2 mediated because if we repeat this experiment with these brain slices using a D2 antagonist, now these two that are different here are not here. So we block the quinpirole effect we block the tonic effect that's there at steady state with this D2 antagonist. And sulpiride then brings back the mutant level of surface dopamine transporter back down to wild type levels. So again, we've got different pathways to consider. I've only shown you this data from the dorsal striatum. That's where we were making our prep. That's where we did our microdialysis studies. So we now went to the ventral striatum. So here again, that's the dorsal striatum data for comparison. Here's the ventral striatum. There's no basal effect, and there's no effect of quinpirole. What these data suggest is that, in fact, in the ventral striatum, the D2s do not control the dopamine transporter trafficking. That is not, they're not hooked up to do that. So that was a surprise to us, and that wasn't in the literature anywhere. And the only reason we found it is because we wanted to find out what was going on with this mutation. Let me show you some other data. I didn't emphasize this, but this D2 receptor to cause trafficking of dopamine transporters does through a kinase cascade that results in the phosphorylation of the dopamine transporter at threonine 53, among other sites. So this is an easily monitorable site because there are phosphospecific antibodies that detect that phosphorylation. So we went in an IP blot where we IP with antibodies against the phosphorylated transporter and then blot for it. What you see is that in the wild type case, with quinpirole stimulation, you can get very nice phosphorylation of the wild type dopamine transporter. But in the mutant, what you have is an elevation already of phosphorylation of that site, and again, it doesn't respond to the agonist. That's in the dorsal striatum. In the ventral striatum, doesn't matter. Those D2s are just not coupled in the same way to the trafficking of the transporter, and we think perhaps mediated through that phosphorylation site. Now, how that phosphorylation site actually then leads to trafficking of that transporter probably involves changes in the cytoskeleton and protein interactions with the transporter that stabilize it at the cell surface, but we're not yet there with that understanding. Now, the last thing we wanted to do actually goes back to that PET imaging study I showed you where the levels of the dopamine transporter by PET in ADHD subjects was reduced. And I told you that that could be there because the dopamine transporter levels were reduced or because there was an elevation in extracellular dopamine and the PET ligand just can't get there. We thought we had an interesting opportunity to test that. And so, the model here would be, this would be our ligand, or it could be even our exogenous dopamine. And if we had a way to discriminate these two, we could show that. And the technique that we used was a version of this amperometry approach that I mentioned before, where we stuck electrodes, carbon fiber electrodes, into the dorsal and ventral striatum. And then we puffed with a micropipette a little bit of dopamine. And the idea would be there would be either little here and a lot here. And so the exogenous dopamine we add should have a change in its clearance kinetics because the dopamine transporter is already in an efflux state, and it's got a lot to do besides pay attention to this extracellular dopamine. And the data came out along those lines. So here is the recording. Remember, I showed you that at the beginning where in a dopamine transporter knockout mouse, if you knock out dopamine transporter copies, you see this prolonged clearance rate. And the same thing you see here, that we puff in dopamine, and in the wild type, it's cleared out nicely at this rate. But in the VL559 mice, you see this. And so the clearance rate, even though the surface expression is higher, and if you wash away the extracellular dopamine, there is no change in uptake of dopamine. In the tissue, in vivo, the clearance rate is now slowed in the dorsal striatum, but not in the ventral striatum. And so we think that's a consequence of not having this D2 coupling. So you don't have as much of these leaky transporters at the surface. You don't have as much of a cloud of dopamine, and you therefore don't have good competition for the exogenous dopamine we put in. Now, it could be that maybe those D2 receptors in the ventral striatum are just not doing anything. Now, it seems silly. There's plenty of D2 receptors there post and presynaptically, and maybe it's just specific to the dopamine transporter. That appears to be the case because we monitored the ability of the tyrosine hydroxylase activity and its sensitivity to D2 agonist. And in the wild type case, we could easily see nice suppression, whether we're in the dorsal or the ventral striatum. And here we show that in the mutant, there is a loss of this coupling here that we don't see here. So what it says is the D2s work, but they couple differently to these downstream effectors depending on the projection of dopaminergic neurons. So we have to be careful about interpreting the effects of mutations and say that all dopamine neurons are alike and all dopamine-related mutants are going to give the same effect. And that was a pretty interesting lesson to come out of this. But I think it has bigger significance. In this particular case, these individuals had either hyperactive predominant or a combined form. So in both cases, they have hyperactivity, impulsivity. They're not in the impulsivity only group. And one possibility is that the manifestation of their traits is driven by this preference of the system, not of the mutation, but of the system, to use this D2 receptor to couple to DAT only in the dorsal striatum, and an area that is subserving control over things like activity and impulsivity. So just to summarize then, we think that in the dorsal striatum, we've got this coupling of presynaptic D2 receptors shoving leaky dopamine transporters into the membrane, compounding a problem that then leads to constitutive suppression here. It's also suppressing dopamine release. I showed you that in the SLY studies early on. And so the net consequence of this is a major shift from excitation-coupled secretion of dopamine to a leak and a tonic level of extracellular dopamine. And those of you who are computational might find it interesting to think about the kinetics of a modulator like this in a circuit and help us understand how it's leading to changes in behavior through one circuit and then not in another. Now, the last thing I'll say is just some food for thought for going forward. We've just begun studies with female animals. Now, there's a male to female bias here, and we thought that perhaps the females would be protected from this mutation. It's actually a little bit more interesting than that. Here is just one set of studies, and we've actually completed 3 or so of the ones I've shown you, but in females. This looks at trafficking of the dopamine transporter. Here's the male data I showed you before, where we have this elevated elevation with quinpirole and a tonic elevation of surface expression going from the wild type to the mutant. If you look at the dorsal striatum of females, there's really no difference here in the response of these animals to this mutation. the females, in the dorsal striatum. They don't show this elevated surface expression, and they don't respond to the D2 agonist. But you go over into the ventral striatum, and now you see this. So in the females, the D2 looks coupled to DAT in the ventral striatum, whereas the male D2 is coupled to DAT in the dorsal striatum. And for someone with a problem with their dopamine transporter, our prediction is they're going to have behavioral manifestations that are dorsal striatal dependent, whereas for the males, but for the females, it's going to be driven by perturbations of the mesolimbic accumbens projection pathway. And so, what would that be? Well, that might be a set of traits that doesn't even look like ADHD. That could have a reward syndrome. It could lead to addiction. It could be any number of other things. But maybe this is one way you get to sex bias, is that you're not protected. You actually have a problem, but your problem fades out of that diagnostic category and shows up somewhere else. So I think it'd be interesting to look at families where you have an affected male and ask about the sister in that family. And it's going to be hard to do. You got to have it, be great if you could find mutants. These things are pretty rare, so I doubt I'm going to find the sisters of these mutants, but maybe we could just look generally and ask that question and ask whether or not there are sex biases in traits that are seen. They're both of them disruptive. But we'll study this a little bit more and see if we can understand something about the sex dependence of the architecture, both at the molecular level and circuit level. So in conclusion, the region-dependent differences that we see in D2 coupling to DAT dictate the penetrance of this mutation with respect to DAT trafficking and dopamine efflux, and they caution us about generalizing to the sites of effects of inherited mutations. We have to know what pathways we're talking about, when the mutant's expressed, and how it actually functions in C2. This inherent dependence of D2 coupling to DAT likely then sculpts the specific dopamine-dependent behavioral deficits that one sees in neuropsychiatric disorders. And you can really pick this up if you monitor the sex differences in your system, in our case, D2 regulation of DAT. And I think if we had found this just asking that question, it would have been an interesting story. But in combination with this mutation, now we have a little bit of an interesting twist to it that takes us down a pathophysiological route as well. So with that, thank you for coming to my talk and hosting my visit to your beautiful institute. I hope you all feel, very proud of it. It's wonderful, and I know you'll all do great work here. These are the people in my group and collaborators that have helped me do this work. So thank you very much.